Let's walk through the microscopic anatomy of our cardiac muscle. Now, when we look at cardiac muscle underneath the microscope, there are some things that you should remember from Anatomy 1, things that you can see. One thing that I'd like to point out are these intercalated discs. So remember, these are these dark bands. They're junctions between cardiac muscle cells that help to anchor those cells together so that they don't pull apart when the muscle's contracting, but they stay together. So here's a picture. We're going to look at this image here on the top left. This is cardiac muscle under a microscope. And you can see really well here, these are intercalated discs. So they're holding those muscle cells together. Even when the muscle cell contracts, it'll stay anchored to the surrounding cells. Other things that we'll find within our cardiac muscle cells is some linker proteins called desmosomes. These are doing something very similar. They are holding those cardiac muscle cells together, preventing them from separating during a contraction. So in this image on the left, we can't see desmosomes here under a microscope. And so over here on the right, this is showing you a desmosome, what it looks like. So the squiggly line is basically the junction between two cells, but you can see these desmosomes, those teal structures holding the cells together, keeping them anchored together. And then lastly, we also have gap junctions, and gap junctions are just like what they sound like they are. They are proteins that have holes in them. And so it allows two cells to pass things back and forth between them. Typically, it's going to be ions that can pass. But in this case, it also allows cardiac muscle cells to be electrically coupled to each other. So if one has an action potential, the one right next to it can have an action potential. And this is important because it allows the heart and all those cells to behave as a coordinated unit. So the heart, all those cells will contract at once, okay? And so we can see here, these are our gap junctions, and you can actually see the little hole in between. So again, they're gonna let things pass back and forth between two different cells. Now, the image on the bottom is showing you some other structures, some of the same structures, right? We can see the intercalated disc here, helping to hold those cells together. We can see gap junctions, and we can see desmosomes, again, still holding those cells together. Now, I want you to notice where those desmosomes are located here. They are actually located within the intercalated disc. So that's why those two structures do kind of the same thing, hold the cells together when they're contracting so they don't pull apart. Now, in terms of what cardiac muscle cells look like under a microscope, they look kind of similar to skeletal muscle cells. They're long. They are striated, so you can see all these little striations up here. Okay, you can see the same thing down in here. You can see the striations. We can also see that cardiac muscle cells are very branched. So I think being able to see the branching down here in this artist rendering, it makes it really easy. You can see all the branching. And if you look up here, you can see the same thing. And then cardiac muscle cells are a little different because they also have one nucleus. They're uninucleate. So there is one nucleus per cell in cardiac muscle, unlike skeletal muscle, which is multinucleate. And then cardiac muscle, we know, is involuntary. You don't have to think about it for it to contract. So in reality, I've mentioned a couple of similarities and differences already, but let's talk about some of these in a little more detail. A few other similarities that we might find between cardiac muscle and skeletal muscle. The sarcomere arrangement between the two is very similar. So y'all should remember what that term sarcomere means. Remember, it is a contractile unit, runs from Z disc to Z disc. So let's go back. We talked a lot about sarcomeres in Anatomy 1. So here we can see this is a Z disc here. Here's another Z disc. Okay, and then we can see the thin filaments in blue that are made of actin. Use my pointer. And then we can see the thick filaments in red down here made of myosin. Okay, so remember the sarcomere arrangement is Z disc to Z disc. Okay, and then we have what's called I bands on the ends. And the I band, I always remember I for light. It is light in color under a microscope because in the eye band, all you have are these thin blue filaments made of actin. Okay, and you can see that on both eye bands. The A bands are kind of in the middle of the sarcomere, and I remember A for dark, has an A in it. And an A band is dark in color because it has the thin blue filaments and also the thick red filaments made of myosin. And because it has both of those, it looks a little darker in color under the microscope. So the arrangement of a sarcomere inside of a cardiac muscle cell looks pretty identical to what we find in skeletal muscle cells. Now, the other thing, if the sarcomere looks identical, then it's going to contract the same way using this sliding filament model. Remember, the sliding filament model basically says that these Z discs, when the muscle contracts, the Z discs are going to push towards the midline. So everything gets pushed in towards the middle and the sarcomere gets shorter. Okay, there's more overlap between these blue thin and red thick filaments. Okay, so this just kind of pushes towards the middle. You can see the blue here. It has room to move in towards the middle, in towards this line in the middle. Okay, so the sarcomere just gets shorter. And those filaments slide over each other. That's all the same between cardiac muscle and skeletal muscle. So then how is it different? Cardiac muscle is very different from skeletal muscle. One has a lot more mitochondria. Remember, mitochondria are where energy are created. So remember this equation, which is carbs plus oxygen forms CO2 plus water plus energy, ATP. Okay, so remember that is cell respiration, and this process happens inside mitochondria. So cardiac muscle cells are going to have a lot more mitochondria so that they do not fatigue as quickly as a skeletal muscle cell would. Okay. Now, cardiac muscle cells, they don't have terminal cisternae or triads. You may or may not remember what that is. So let me pull up a little whiteboard. So let's look at skeletal muscle, right? If I were to draw out skeletal muscle phys, I would draw out my neuron. Okay, and here is my muscle cell. It's really long. And my muscle cell has T-tubules and terminal cisternae, which are portions of the sarcoplasmic reticulum. Together, those are called triads. And then we also have our sarcomere, Z disc to Z disc, with those thick filaments in the middle. Okay, so if I were to draw this out like skeletal muscle physiology, that's kind of how I would draw it out and walk through muscle physiology. Now, cardiac muscle cells, they do not have terminal cisternae, which are these enlarged portions next to the T-tubules. Now, together, a terminal cisternae on either side and the T-tubule are called triads. Well, if they don't have terminal cisternae, oh, that's not letting me erase. Well, if they don't have terminal cisternae, then they can't have triads. Okay, so we'll take those off. All right, so let's go back. So they don't have terminal cisternae, and because of that, they also don't have triads. The cells, like I mentioned, are branched and they have one nucleus. The other thing is our cardiac muscle has a very unique type of troponin. Now, remember troponin and tropomyosin. These are regulatory proteins. They sit on actin and they prevent myosin from latching onto actin unless you're ready for a contraction. Because remember, that gets back to the sliding filament mechanism. When a sarcomere gets shorter, let's go to this picture. These red filaments made of myosin have to latch on to the thin filaments and they have to push. Okay. Well, those thin filaments made of actin, they have proteins, regulatory proteins that cover over their active sites. I think of them as like the chastity belt for muscle contractions. And those are called troponin and tropomyosin. And so cardiac muscle actually has a very specific type of troponin. And typically when heart muscle has been damaged, the levels of troponin go up. And so they can do a blood test and identify if you have elevated levels of this very particular type of troponin, then chances are you've had a heart attack or you've had some kind of heart damage. A couple of other differences is, remember, cardiac muscle is involuntary, but it does have what we call autorhythmicity or automaticity. I like autorhythmicity. And this means that it basically sets its own rhythm. You actually have a pacemaker in your heart that sets a pace, a sinus rhythm of about 75 beats per minute. So normally, let's go back to our whiteboard. Normally, if we think of skeletal muscle, right, which is up here, in order for a skeletal muscle to contract, remember it's under voluntary control. So it has to have stimulation from a neuron. So remember, an impulse comes down a neuron. Neurotransmitters would get released into the synaptic cleft, and those neurotransmitters would bind to receptors on that motor end plate, and sodium channels open, and that's going to depolarize the membrane, right? So that's normally how we start muscle physiology, skeletal muscle physiology. It always starts here with an impulse coming down a neuron. And it's got to jump the gap, and sodium channels have to open for it to continue through your skeletal muscle cell. Cardiac muscle cells. These sodium channels within our cardiac muscle are leakage channels, meaning they're always open. They're not reliant on a neurotransmitter. So cardiac muscle cells do not need a neurotransmitter. They do not need nervous impulses, which in some ways is really great. So this is how people can have heart transplants. We can take a heart out, put a new heart in, shock it, get it into a normal sinus rhythm, good to go. Remember, if they sever nerves, they can't reattach those and regain function. Let's go back to our screen here. So you don't need nervous stimulation for a heart to contract because all those sodium channels are leaky. And you have basically in your heart, you have a pacemaker that's going to set a rhythm for the heart. And so what we say, all your heart muscle cells are interconnected. So the fastest cell or group of cells to depolarize is the one that's going to set the pace. So the fastest one to depolarize and generate an action potential is the one that's going to be setting the pace. Okay. And it'll spread throughout the whole heart. And we'll talk about that in a little bit. All cardiomyocytes, all heart muscle cells will contract as a unit, or none of them do. And again, this gets back to those gap junctions we were talking about. And that's really important. When your heart contracts, how it contracts is atria contract, then ventricles. Atria, then ventricles. You want all of those cells around the atria to contract at once. You want all the cells around the ventricle to contract at once, in order for your heart to be effective at moving blood throughout your circulatory system. If heart muscle cells start contracting individually, it makes the heart non-functioning. It almost looks like the heart is quivering, looks like the cells are quivering instead of that whole chamber contracting. You have sort of a quivering heart, and we call that fibrillation. And so this is when they take those paddles and the electrical paddles, and they rub some conducting jelly on them and put them on someone's chest and yell clear and shock the person. They're trying to shock them back into a normal sinus rhythm. We call that defibrillate. Okay, so cardiomyocytes contract as a unit, or they don't contract. And the other thing, the last thing I have listed down here is that your cardiac muscle the cells have a very long absolute refractory period. That term should be familiar to you. We talked about this in muscle physiology in Anatomy 1. The absolute refractory period is the time when a muscle cannot generate another contraction. It cannot. In heart muscle, the absolute refractory period is long. And the reason is you want to give the cells time to relax back out so that the chambers can fill up with blood before it contracts again. That is a long absolute refractory period, letting the cells relax so that those chambers can fill up with blood before all those cells contract and the chamber ejects the blood again. And so that's really important, but also having this long absolute refractory period prevents tetanic contractions. Again, that's a term from Anatomy 1, tetanus. Tetanic contractions are when a muscle contracts and it stays contracted. So you can imagine in the heart, if all those muscle cells were to contract and stay contracted, then those chambers would not be able to relax and fill up with blood. So basically your heart would stop functioning. So we need a long absolute refractory period to allow those chambers to relax out and fill up with blood and prevent tetanic contractions, just constant contractions. I mean, you can imagine if the muscle cells had a short refractory period, they could squeeze all that blood out. And if they relax a little and squeeze and relax a little and squeeze, they're still not filling all the way up. So circulation will be lowered. So again, you got to have a long refractory period. Now, in terms of the energy requirements for cardiac muscle, it has a lot of mitochondria, which again, remember, mitochondria is where cell respiration happens. Carbs plus oxygen, oops, my plus doesn't look like a plus, forms CO2 plus water plus ATP energy. Okay, so that's cell respiration, and this happens inside mitochondria. So cardiac muscle has a very high dependence on aerobic respiration, meaning you have to have oxygen. Your cardiac muscle does not do well without oxygen. However, cardiac muscle can very easily switch fuel source, which is here. So you're not eating carbs? Okay, your body's going to burn lipids with the help of oxygen to make energy. No problem. You don't have any lipids, somebody's starving, burn proteins. Your heart muscle can even burn lactic acid to create energy. So the greatest danger to a myocardium is from a lack of oxygen because it relies on oxygen. It's not from a lack of nutrients. Someone can be starving, but as long as they're breathing, their heart will continue to function because it can switch fuel sources very quickly. Even if that means your body has to consume your own muscle tissue, you've got some fuel in there that can be used to create energy for your heart to continue to contract. Now let's mention a little bit about some imbalances, some homeostatic imbalances with oxygen supply. Anytime oxygen supply gets low going to your heart, we call that ischemia. Now, in reality, that term ischemia, you've all had ischemia before. Your hands ever fallen asleep? Your leg ever falls asleep? It is truly when you block blood flow to an area, you are blocking the oxygen supply to the area and your nerves are tingling, which is why you get that feeling. Now, ischemia to a limb isn't a huge problem because we have other channels for blood to pass into those tissues. But ischemia in places like a really active organ like the heart is a huge problem. So what's going to happen is anytime you block oxygen flow to the heart muscle itself, those cells will have to go through anaerobic respiration, which we said they don't do very well. And remember, anaerobic respiration produces lactic acid. That's one of the byproducts. Now, lactic acid is an acid, which means it has a high hydrogen concentration. This is going to cause calcium levels in the cells to go up. That's going to damage the mitochondria. And without mitochondria, you can't produce enough energy for your heart to keep working. Also, high acid levels, high hydrogen ion content causes gap junctions to close, which means the cells can't communicate, they can't send signals back and forth, they cannot electrically couple themselves, and that can lead to fatal arrhythmias. So this is why if you block blood flow to the heart muscle itself, It's so damaging to the heart. So we talked about how myocardial infarctions are heart attacks, and this is when you're blocking blood supply um, to the heart muscle, and how it can really damage the heart muscle or even lead to death. This is physiologically what's happening when you block blood flow to the heart. So you go through anaerobic respiration, you produce a lot of acid, which causes you to pump out calcium, which kills mitochondria, so you can't make energy, and then it causes gap junctions to close, so cells can no longer contract at the same time. That's a big deal, okay?